Isolating and screening bacteria and fungi found within mangrove soil in South Florida in search for accessible antimicrobial natural products. Each mangrove ecosystem around the world houses a unique microbiome within its soil. Many studies on different mangrove soils in countries around the world have identified organisms that produce antimicrobial natural products, but no research has ever been conducted on South Florida's mangrove soil microbiome. Secondary metabolites are organic compounds produced by organisms that aren't directly involved in growth, development, or reproduction, but often play crucial roles in survival. These molecules synthesized within cells include a vast array of compounds like alkaloids, terpenoids, and phenolics. Natural products refers to these secondary metabolites when they're harnessed for medicinal, agricultural, industrial, or commercial purposes due to their diverse biological activities, such as antimicrobial, antioxidant, or pharmaceutical properties, making them valuable resources for drug discovery, agriculture, and various industries. In the chart above, you can see how the release of secondary metabolites from organism A caused the lysis of organism B. This is also known as antagonism. My question is, can the microorganisms isolated from South Florida mangrove soil be used as a source of new natural products? So first, in order to isolate the organisms that I obtained from the mangrove soil samples, I had to follow the protocol which is shown here. I gave each organism an ID based on the agar that it was grown on and the order in which it was named out of all organisms found on that particular agar. Once I had the organisms isolated, I then proceeded with a secondary screening procedure to identify the organisms that had antimicrobial characteristics. I performed an antagonism/dual culture assay, and the procedure is shown on the screen. After completing my antagonism/dual culture assay, I found that organisms B6 and E13 had antifungal properties. Here you can see the zones of inhibition that each organism created outlined in red. The zone of inhibition is where the model organism, Aspergillus niger, wasn't able to grow. I decided to extract the secondary metabolites from organism B6 and test to see their antifungal potential. Here you can see that the metabolites extracted from organism B6 did show antifungal activity as a zone of inhibition, which was again outlined in red, was created around the metabolite holding disc. Once I had performed the primary and secondary screening on the organisms obtained from the mangrove soil, I extracted the DNA from the organisms of interest and primed them for sequencing. For the organisms that phenotypically resembled fungi, I primed them with the ITS-1 primers, as the ITS region is the most diverse genomic region amongst all fungal DNA. In the chart shown, the quantified primed DNA concentrations can be seen. Organism B6 has a significantly lower concentration of primed DNA, which could be a signal of it being an organism other than a fungus. The reason for this is because if the DNA isn't able to be primed with the ITS primers, then it won't be able to be amplified during PCR, resulting in a low DNA concentration. This is most commonly seen amongst organisms that don't have the ITS region in their genome. And here it can be seen highlighted in red that the previous explanation is justified by the results of organism B6's read on the sequencer. No organism was able to be classified as the lack of priming on the DNA prevented the DNA library from sticking to the flow cell on the sequencer. If the DNA isn't stuck to the flow cell, then it can't be read. Here you can also see the data from the other organisms that were primed with the ITS-1 primer. Similarly to the ITS-1 primed organisms, phenotypic traits were used to decide the organisms that would be primed with the 16S primers. The 16S region is found in bacterial DNA and is the region in which the most diversity is found. It is one of the most commonly sequenced regions as it plays a major role in bacterial identification. Based on the chart above, all organisms seem to contain the 16S genomic region. I decided to prime the organism B6 with the 16S primers as well, and it can be seen that it had a significantly higher DNA concentration than that of when it was primed with the ITS-1 primer. This helps with the surface level differentiation between what is bacteria and what is fungi. Here you can see the identification of the organisms that were primed with the 16S primer. In conclusion, 23 organisms within South Florida mangrove soil have been identified, with 7 unique fungal species, 2 unclassified fungal species, 3 unique bacterial species, and 4 unclassified bacterial species. Also, it has been confirmed that antimicrobial products can be found in South Florida mangrove soil. In the future, I would like to refine the antifungal products of the isolated organisms and create usable antifungals and isolate more organisms with potential antimicrobial characteristics. Thank you.